All right, thanks everyone for being here, and thanks to LGC for setting this up. That was a tough talk to follow, but I'm going to try. So yeah, I'm going to talk today about our lab's efforts to utilize targeted SNP-based genotyping to advance our work in cotton introgression. Broadly speaking, if you're not aware, introgression is just the transfer of germplasm from one species into another through the production of a hybrid. In our case specifically, we're talking about introgressing non-commercial germplasm into a commercial cotton background. The reason we want to do this is cotton is facing a number of different challenges, effects of climate change, the overuse of soil and water resources. Some of these are common to any agricultural system, some are unique to cotton. I'm not going to go into too many details on this, you can kind of use your imagination, but one I would like to highlight is a fusarium wilt. There's a variety FOV4 that's currently spreading across all the cotton producing regions of the United States. And where it occurs, it causes total economic loss, and the commercial lines have essentially no resistance. Which brings us to the heart of our problem. We're trying to face all these challenges with germplasm that lacks the diversity necessary to face these challenges. So we're going to have to think outside the box a little bit, and we're going to bring in germplasm from wild species and niche cultivars and maybe underused cultivars. And fortunately for us, these between them represent a rather large reservoir of germplasm that we can readily take advantage of. Okay. The cotton species originated as a diploid, and then about 1 to 2 million years ago there was a polyploidization event, and that led to the creation of at least 5 tetraploid species. Gossypium hirsutum in the top right corner there, that's the commercial line. 95% of the fiber production, give or take, throughout the world. If you're wearing anything made of cotton, chances are it came from this. And this is the one that has been intensely selected for superior production traits and now lacks a lot of genetic diversity. Barbadense, the one next to this is one that has been selected for superior fiber qualities. If you own anything made with Egyptian cotton, unless it was fake advertising, this was made by Barbadense. And then you also have tomentosum and mustulinum. These are non-commercial, they have no production value, but they do have disease and stress tolerances that we'd like to utilize. Okay. So there's a variety of different things you can do with introgressed populations, and depending on what your final goal is, your breeding scheme is going to look a little bit different, but they all start with a cross, in our case, between a commercial hirsutum and one of those 3 donor species that I mentioned. Take that to produce an interspecific hybrid, and then you can produce, through repeated back crosses, a variety of different populations. In our case, we're primarily focused on chromosome substitutions and chromosome segment substitution lines. So a chromosome substitution line, very simply, it has a single chromosome inserted from one of the donor species against an otherwise commercial background. And the reason we do this is if you isolate that chromosome against a commercial background, any of the phenotypic differences you can link back to the introgressed material. The goal is to create one of these CS lines per chromosome per donor species. So the way we go about creating these, same as any introgression, you start with the 2 parents, the commercial hirsutum and one of your donor species. These are a little bit unique in that the commercial line that we're using has a single copy of one chromosome missing. In this example, it's a chromosome 4. This is known as an aneuploid. It is otherwise a normal hirsutum line, with the exception of having only one copy at this loci. So we cross that, and then look for the interspecific hybrid that has inherited the missing copy. This is what we can use for developing these lines. Unfortunately, most are not going to look like that. The vast majority are going to be normal heterozygotes, normal ploidy level, and are not particularly useful. So this necessitates screening very large numbers of seed to identify this rare individual. Historically, the way we've screened has been to do a phenotype screening in the field, look for the aberrant phenotype, and then do chromosome preps to verify it in the lab. The downsides of this are phenotypes are not always obvious and can be masked by environmental factors, and chromosome preps are time-consuming and difficult. And in both cases, you have to take all of your seeds, grow them to maturity to identify the one or two that you actually want. So with that in mind, we were looking at a marker-based alternative. Reasoning being any marker that can discern your donor from your commercial line that's localized to your target introgressed chromosome is going to be homozygous here. So all of the ones that we want, all the rare genotypes are going to cluster with the donor. All the heterozygotes can be discarded. So once we've selected the interspecific hybrid that we want to use as a parent, we cross that back to the original aneuploid, and then again screen the back cross generation to find the rare genotype once more. And we take that one and cross it back, and we do this many times till about the BC5 generation usually. And the reason we're doing that is with each back cross, you're eliminating 50% of the donor genome from the non-target chromosomes, whereas the introgressed chromosome, since it has no pair, it cannot recombine, so it remains intact. At this point, you can take the BC5 generation, self it to produce a euploid, normal ploidy level. And this is a true breeding line that we can use for trait discovery, phenotype mapping, and developing chromosome segment substitution lines. It is almost entirely hirsutum, commercial background, with the exception of the introgressed chromosome, which is intact donor. So the CSSLs, they're a similar idea, but you're taking the chromosomes and you're breaking them apart into segments. And if you get enough of these overlapping segments, you can get full genome coverage. The advantage of this is that it allows more specific phenotype to genotype mapping, and it lets us work with populations that are difficult to develop as CS lines. Downside is that you have to have a much larger population to get good coverage, so that necessitates even more screening. Okay. So the pipeline we're really looking at to do this efficiently is to have some sort of genotype screen in place that we can do quickly. We have seasonal growth requirements. We need to collect the seed, screen it, and then plant next season. We need to be able to target different regions of interest depending on the population we're trying to develop, and it needs to be cheap. We're doing thousands of screens every year. Over many years, it adds up. So in practice, this means we need a high throughput DNA method that can be coupled with genetic markers that can be targeted, that can discern the parent, the commercial from the donor and the heterozygote, and that works well with our extraction methods. Okay. So we have an in-house DNA extraction method. It is very simple. It does not require any specialized equipment, a rotary tool and some plates and some buffers that anyone can make. It is extremely cheap and fast. You can do 10,000 seeds easily enough between generations. The seeds do remain viable, which is essential if we're screening for the next generation. And the plated DNA can be dried and stored at room temperature pretty much indefinitely. The downside is this is a crude prep, so there are certain assays it does not work well with, but we found that it works just fine with most PCR-based assays. So with that in mind, we've been developing KASP assays for use on screening these populations. For anyone who's not familiar, KASP assays work by competitive binding and amplification. So you have your two forward primers. They contain one or the other SNP. They also contain a unique oligo tail at the 5' end, as well as a common reverse primer. Then you have your KASP master mix. These contain oligos that correspond to those tails, and they've been labeled with a fluorochrome, either FAM or hex, and an associated quencher to prevent fluorescence, as well as your enzyme buffer salts, etc. So when you combine those in the presence of template DNA that contains a SNP, the appropriate forward primer will bind, amplify, When it does that, the tail is part of the product, which then incorporates in subsequent amplification steps the oligos, which causes unquenching, and then you can read the fluorescence and determine the presence of one SNP or the other. Okay. One of the things that appealed to us about this is it does not require a labeled probe. That makes it a lot simpler for our applications, especially if you're going to be testing and discarding hundreds of different assays. It also lets us work with smaller genomic regions. We don't necessarily have a lot of genotype information to work with, and it simplifies the primer design. Okay. The equipment is also very basic. We didn't have to buy any special equipment for this, just a thermocycler and a fret-capable scanner. Pretty much any lab would have that. And the software, very simple. You don't have to program or have any particular knowledge to be able to use it or do the analysis. Okay. The SNP markers that we're using came from the cotton SNP 63K array that identified about 18,000 interspecific SNP, along with some flanking DNA sequence and a linkage map that ties each SNP to a location on the chromosome. We designed primers using batch primer 3, added the appropriate tails, and then ordered our oligos from IDT. Okay. So the goal we were going for here was about 15 SNP assays per chromosome. We figured that would be about adequate for most of our needs, with a spacing of 1 every 10 centimorgans. That would let us determine with relative accuracy where chromosome breaks occur. And just to keep it simple, we're focusing only on biallelic SNPs that discern between the commercial and all 3 of the donor species. There were triallelic SNPs and SNPs among the donors, but for this purpose, we're not interested in those. Okay. The SNP assay validation was very simple. We just compared it against the 3 different parents and just looking for very clear clusters with no ambiguity between the different genotypes, and then no amplification, of course, and the no template control. Okay. So to date, we have designed and validated 413 of these at 382 unique locations. That's just under our goal, but it's pretty close, of 15 per chromosome. Average gap of right around 10 centimorgans. There's still a few areas where we're interested in designing a few more for better coverage, but right now this gives us a pretty good library to start with, so we can select which ones we want for any particular assay for any particular population. So currently we're using this for the CS and CSSL line development. For backcrossing and inbreeding, you can use it to determine heterozygosity or homozygosity and selectively maintain or discard those populations, depending on which ones you want. There's a project looking at transmission rates, and then of course you can also use it for purity analysis. At what rates are we selfing versus outcrossing, and breaking up genomic blocks and identifying rare genotypes that might otherwise be difficult to identify. Okay. So just kind of a wrap up. If doing this efficiently, introgressing germplasm, it really helps if you can have a targeted genotyping system that you can utilize. And we found that the KASP chemistry is fairly simple and relatively inexpensive, and it works well with our low quality DNA preps. And with our library of assays now, we can target whatever we need for any particular population. And this has really greatly improved our efficiency and cut back on costs. For instance, last year I was able to partially implement this. We were still working on the development stage, but I was able to screen 2,000 of the seed before going into the field and discarded 96% of them. So that significantly cut down on the field space and the manual labor necessary that year. And it's also given us some research opportunities that might otherwise have been just impractical due to cost and time. So that's it. I'd like to thank my lab and A&M AgriLife and Cotton Inc. for the funding, and then LGC for organizing this workshop.
Questions for Robert? how do you maintain these aneuploid lines? And like, what's the genetic stability of them?
Okay, so the question was, how do we maintain the aneuploid lines? So the aneuploid lines, they're kind of spontaneously, they arise spontaneously due to whatever reason. You can create them with chemical or radiation treatment, but most of them, the ones we have, just arose spontaneously. And we maintain them through cuttings. You can self them and get self-seed, and some of the self-seed will have the aneuploid status, but it's going to be the same as the problem I mentioned before, where it's going to be a very small number. So it's easier to just keep the plant alive. And yeah.
Okay, we have one question from our Slido from the audience, I think from Manoj again. So how did you make sure that the gene of interest regulated trait targeted is coming from the chromosome that's introgressed from the donor parent?
Right, okay, so because this is, the chromosome is introgressed into an otherwise uniform commercial background, so the assumption is that any differences are going to be due to that introgressed chromosome. Now, there are going to be issues with like gene by gene interactions, so we don't necessarily know exactly that every trait comes from that introgressed chromosome. It might be due to a gene combination. And so that's where the CSSLs really help, because if you can break it apart, it lets you understand that mechanism a little bit better. But at least the CS lines give us kind of a jumping off point.
Okay, thank you, Robert. Thank you.
Thanks.